Hello. So, welcome everybody to our ICCUB colloquium. Before we start, I want to make a short announcement that we are going to have one more colloquium by the end of the year. This will be in December and will be motivated by the Nobel Prize. So Maciek Levenstein will come and give us a colloquium about the physics of the Nobel Prize. We still don't have a title or abstract, but I want to make the announcement because this colloquium will be on a Wednesday because of a schedule problem. So it will be Wednesday, December 13th. So I just want you to remember that the next colloquium will be on a Wednesday and it will be about the Nobel Prize Physics. And so please check for the announcement whenever it's ready. And now we will have a colloquium today, and I will let Jordi Miralda introduce our speaker.
Thank you, Jorge. I'm very pleased to introduce to you our visitor today and colloquium speaker, Professor Antonio Lazcano, who comes from Mexico, from the El Colegio Nacional and Universidad Nacional Autónoma de Mexico. Well, he already obtained his PhD there at the UNAM. And then throughout his life, he has been dedicated to the research in evolutionary biology, the early stages of the evolution of life, prebiotic chemistry, and of course, the emergence of life in the primitive Earth. So he has also been doing research on meteorites and simulation experiments on the primitive Earth and how life emerged in those conditions. He has been visiting numerous institutions and with a lot of collaborators in many countries, in France, here in Spain. He has been coming for a long time to many institutions in the United States, in Russia, in many countries. And in addition to all his research activity, he's also a very good disseminator and teacher. And so he has published a lot of books on disseminating the science of evolutionary biology and the origin of life. A very famous one called El Origen de la Vida, The Origin of Life, that he has sold incredibly well, like 800,000 copies. And also another one that he published about Alexander Oparin. I think the title was La Chispa de la Vida, The Spark of Life. So, this is also very something that is very important, of course, the dissemination of science to the general public, and I think he has excelled on this. He has also collaborated with many famous biologists and been in advisory boards. Among them, even though I know that he doesn't like the term astrobiology, but he has been in the advisory board of the NASA Astrobiology Institute and various places in the United States. He's been president of the International Society for the Study of the Origin of Life, a place where also there were other famous scientists who were president of these societies, including Alexander Oparin and Stanley Miller, of whom he will tell us about today, about this experiment. And also he was strong collaborator of Joan Urey, who is, of course, very important Catalan scientist, and we know that this year we're celebrating the centenary of the birth of Joan Urey. I think it was just a few weeks ago. So we're glad to have him here today, and if you want to ask him any questions about all these topics, he will be here throughout today. And actually tomorrow he will also be giving another disseminating public talk at the Institut d'Estudis Catalans tomorrow, Friday at 7 in the evening, so you're welcome also to come there. Thank you very much, Antonio, and thank you very much for being here. I suppose you already have your microphone.
Right. Thank you so much, Jordi. I'm absolutely delighted to be here with you. I have been many times to Spain, many times to Catalonia. I realize that it is important to make the distinction, but I feel very much at home here. I was very lucky to be introduced to Catalonian science by Professor Joan Urey, who was a friend, a mentor, a very dear colleague. And but through Joan Urey, I was actually able to meet Stanley Miller. And of course, you all know that the Miller experiment was simulation under laboratory conditions of the possible chemistry that took place on the primitive Earth prior to the origins of life. That being said, I think that everybody can recognize the significance of the experiment, the way it was designed, how it is interpreted. But what many people do not know is the story behind the actual experiments. All the doubts that Stanley Miller had, the situation that he had to face when the experiment was published, the issues in publishing, in getting published his first paper. So I will talk a little bit about that because I think it's a fascinating lesson to be learned, especially for young scientists who always feel that, we always told them that just science is always advancing directly, and that is not always the case. So just to put the things in context, yes, what we call prebiotic chemistry is in fact the simulation or the attempt to study either by simulation or by analysis of primitive solar system bodies like comets, meteorites, asteroids, the events that took place prior to the actual origins of life. And all this was proposed by Alexander Ivanovich Oparin in 1924, and then in a second book, which I think was truly a masterpiece of evolutionary analysis, that was published in Russian in 1934 and then translated in 1936. The basic idea behind Oparin's proposal is that life is not the outcome of a sudden event of a spontaneous generation, but rather that it is the result of a number of processes, of events that took place prior to the appearance of the first living entities, which for Oparin, in contrast to what many people were saying at the time, were actually bacteria that were fermenting, there were the organic compounds in the primitive oceans, the same compounds that they had formed them. Now, the book was published in 1936 prior to the recognition of the role of DNA as genetic material, prior to, of course, to the publishing of the double helix structure of DNA. So for Oparin, like many of his colleagues, including in Western countries, in protoplasm, which is that undefined liquid inside cells, both genetic and metabolic processes took place. Oparin assumed, and this was quite extraordinary, he coming from a biological background, he discussed with astronomers, he discussed with geologists, with chemists, and so on, and he assumed that the primitive Earth was in fact endowed with a reducing atmosphere. And the idea of a reducing atmosphere was absolutely consistent with the well-known observation that if you have free oxygen, organic compounds rapidly decompose. All of us have the experience of cutting a potato or a banana in the morning and see how it becomes brown. In fact, the browning of the apples and the browning of the banana is the oxidation of the organic compounds. And it was very accurately described by a chemist whose family name was precisely Browning. So when we speak about the Browning reaction, it's both an homage to this organic chemistry. Now, for Oparin, in this type of reducing atmosphere, you could have the non-biological synthesis, the abiotic synthesis, the prebiotic synthesis of organic compounds. Oparin always refused the idea that living entities were at first originated from one single molecule. So he assumed, following a Hegelian tradition that was very strong in Russia, the idea of droplets of organic compounds. Eventually, he decided to choose quasar as a model of the structures that preceded the first cells, and then you have cells. The book published in 1936 is absolutely extraordinary. A number of people were engaged by reading the book, were absolutely fascinated. But 1936, I do not tell to underline this, in Spain, already the Second World War had started. Few months later, after the book was published, Franco started the coup that led to the downfall of the Republic. And of course, the people had other things to do apart from being interested in the study of the origins of life. So it was not until the war, until after the war, when the people decided to look back at issues like the origins of life. And one of the first to do so was an extraordinary scientist, Harold Urey, who received the Nobel Award for his discovery of deuterium, the deuterated water. He had been engaged in the Manhattan Project, and like many scientists of the time, he was horrified by the results. And he decided to move as further away as he could from science that could be used in military, and the development of military weapons. So nothing is further away from the military applications of science than astronomy, or so we think. So he decided to try to understand the chemistry of the primitive Earth, and in particular, the chemistry of the primitive atmosphere. And his argument was extremely elegant. It's an extraordinary simplicity, but extremely elegant. He said, the primitive Earth, together with the rest of the solar system, was formed by the collapse of an interstellar cloud. Hence, the composition of the primitive atmosphere of the primitive Earth must reflect the composition of the interstellar cloud. And of course, the most abundant element in the universe is hydrogen. So therefore, he concluded that all the chemistry of the primitive atmosphere was defined by the presence of hydrogen. These equations are not balanced, but you can see very clearly what was the argument behind Urey's proposal. Carbon will react with hydrogen and give methane. Nitrogen will react with hydrogen and give ammonia. Oxygen will react with hydrogen and give water. The actual rate of constant rate of this reaction is 10 to the 45. Hydrogen sulfide will react with hydrogen and give hydrogen sulfide, and there will be very little CO2. Urey published his model of the primitive atmosphere in 1952 in a paper in the Proceedings of the National Academy of Sciences. At the time, he was already distinguished professor at the University of Chicago, the Department of Chemistry. And when he presented his lecture, when he presented his results, there were two young people sitting in the audience. One of them was Carl Sagan, the astronomer, an extraordinary promoter of planetary astronomy, who was very much in his style, sitting in the front row. And in the back row, almost facing the wall, was Stanley Miller, who had just joined the Department of Chemistry in order to do his PhD. Both Stanley Miller and Carl Sagan told me exactly the same story, that when Urey, independently, that when Urey finished his talk, somebody in the audience raised the issue of the work of a Russian chemist, biologist that had proposed this kind of atmosphere to explain the actual origins of life. At the time, Stanley Miller had just started his PhD thesis with Edward Teller, a famous, very intelligent physicist, but someone you wanted to keep away from. He was the father of the so-called hydrogen bomb. He was a collaborator with the CIA, with the State Department. It's not the kind of person you will invite to your table to have dinner. And Edward very soon left. He was brilliant, by all accounts. Left to Los Alamos to work on the hydrogen bomb. And Stanley Miller was left in the very peculiar situation of being a graduate student without an advisor. So he decided after a few weeks to go to speak to Harold Urey and ask him if he would be so kind to direct his PhD thesis on the simulations of the primitive atmosphere. And Urey said, of course not. And he said, of course not, because you want to give to your students a project that has a significant level of success. And Urey was not quite sure that there will be a good subject for a PhD thesis for a student that has to graduate before the scholarship ends and has to find a position and so on. So he advised Stanley Miller to do something else. Stanley persisted on the idea of the primitive atmosphere, and knowing well both of them, I can imagine the intensity of the discussion. There were like two people from Sicily or from the Basque Country discussing. It was really very, very strong personalities. So after a while, they came to an agreement. Urey said, okay, I will give you 6 months to design an experiment and see if you have any results. If you do have any preliminary observations that are conductive to a possible PhD thesis, then you can go on. So Stanley Miller was encouraged with this idea and then decided to actually design an experiment. And here you see the experiment, which is very well known. 2 flasks connected with these tubes. Here you had boiling water. The water vapor came out and mixed up here with methane, hydrogen, and ammonia. The mixture was subject to the action of spark discharges for about a week. And then when the mixture cooled down, you could just collect the material from down here and analyze it. The actual experiment was very small. You can see here the upper flask, and it's about the size of the head of a graduate student. But the picture also tells you a little bit about the conditions under which the people used to work at the time. The upper flask was surrounded by asbestos, amiant, which is something that no one in his right mind will use today, but those were the times, different times as of today. And you can see the way the students dress up to work in the lab. White shirt, tie, cufflinks, and so on, which is slightly different to the way that not only students, but also professors dress now. So Stanley proceeded with the experiment, and here you see the experiment at point zero. Many of us, many times asked Stanley Miller what happened to the original apparatus, and the answer was always the same. It was very difficult to clean it up, so I ended up throwing it away. So a classical experiment ended up in the dustbin of the Department of Chemistry of the University of Chicago. But Miller left such a detailed instructions of how he had done the entire experiment that it's quite easy to replicate. And these photographs actually come from a replication of the original experiment done in the laboratory of William Schoff at the University of California, Los Angeles. And here you actually see the experiment at point zero. You can see here the spark flying between the two electrodes. And this actually shows that the spark is flying from one electrode into the water that has accumulated on the second electrode, which will be somehow equivalent to have an electric discharge falling upon running water or a small pond, a small lake, or the ocean, which is something we actually see. Well, after 5 hours, only 5 hours, you can see how the inner contents of the upper flask have changed very clearly. You see a dark reddish polymeric material going down quite dense into the lower flask, and you actually can see how efficient the experiment is in changing the inner composition of the apparatus. So Stanley Miller was absolutely thrilled. He made a paper chromatograph of the results, and here you see, given the proper reactants, you can see the spot indicating the presence of aspartic acid, glycine, alpha-alanine, beta-alanine, alpha-aminobutyric. Neither beta-alanine nor alpha-aminobutyric acid are components of the proteins, and a couple of additional compounds that we know now were actually 2 more amino acids. Stanley was absolutely enticed by the results. He showed them to Harold Urey. Harold Urey was very happy and told him, write a paper, and we will submit the paper to Science, which made, of course, Stanley quite happy about it. So Stanley wrote the paper, and as is normal, he put his name first and then the name of Harold Urey, and gave the manuscript to Professor Urey. And Professor Urey immediately erased his own name and told him, you were the one who wanted to do the experiment. You designed the whole work. You wrote the paper. You are the one that deserves the credit, which is a lesson in generosity that we nowadays see very little going on. Nowadays, most of us feel that our colleagues or our students are our potential competitors and not our collaborators, friends of a student. But Harold Urey gave an extraordinary demonstration of generosity in letting Stanley write and send the paper and be the sole author. So the paper was sent to Science, and several of the letters and material I will to show you now actually come from the archives of Stanley Miller and Harold Urey at the University of California, San Diego. They are very generous with the material, and the only condition is to acknowledge the source, which is exactly what I'm doing now. So Stanley sent the paper to Science, and the paper was received in Science in April 6, 1953, and it's the typical message that we used to receive before email. Thank you for your communication on a production of amino acids under possible primitive air conditions. You will hear us later regarding the possibility of its publication. And Stanley was perfectly happy. Being happy with the paper already submitted, the next Sunday, or a couple of Sundays afterwards, he was reading the Sunday issue of the New York Times, which is always a very, very big issue, a very gross paper, and he found a notice that scared him like anything. It was published on March 18, 1953, and this is a transcription. It says that the title of the issue was "Looking Back 2 Billion Years," and it said, in Ohio State University laboratories, Dr. Walland McNevin and his associates are recreating the Earth's condition 2 billion years ago, long before there was life on this planet. This was a general idea. One purpose of the study, Dr. McNevin explained, is to answer this scientific question. Did extreme complexity of chemical compounds develop before life appeared, or was this a result of life processes? Some of the experiments led the researchers to the conclusion that natural heat, light, and electricity could bring about highly complicated chemical reactions before living beings evolved. Artificial lightning was also used because there is evidence, I don't know what kind of evidence, that lightning was almost continuous in the early days of the Earth. 100,000 volt spark sent through an atmosphere containing methane, Mars gas, and water vapor produced resinous solids too complex for analysis. And Stanley almost had a stroke. Almost had a stroke, but this was precisely the experiment that he was working on and the result that he had submitted. So he very rapidly wrote to Professor Harold Urey. This is a copy of the letter that he sent on the very next day. And let me just transcribe, show you the transcription of the first paragraph. Dear Dr. Urey, I am sending you a clipping that I saw this Sunday New York Times. I'm not sure what should be done, as their work is, in a sense, my thesis, which is exactly what can happen to many of us. Many of us have faced a similar situation. And Professor Urey became absolutely infuriated. And so he rapidly wrote sent a telegram to his secretary, Lucille McCormick, who was, you could see, sort of a Medusa-like character. That's the way all the people describe her. And said, send Miller's papers to the Journal of the American Chemical Society, Urey, University of California. Urey was absolutely infuriated. So Stanley retired the paper, took away the paper from Science, sent it to the journal, to JACS, which is a very good journal, Journal of the American Chemical Society. But when he took the paper away from Science, he received a letter, again, this comes from the archives in UCSD, from Meyerhoff, who was at the time the editor-in-chief of Science, and the letter said, dear Mr. Stanley Miller, whatever, although your paper, A Production of Amino Acids Under Possible Primitive Conditions, was submitted to us by Harold Urey, developments of the last few days prompt me to turn to you as the author of this paper to seek your advice and instruction. And then what Meyerhoff offered was to send me, as Stanley, send me again the paper. I will see that it is very rapidly reviewed. And if you agree, I promise to publish it as the lead article in Science. So Stanley Miller called Harold Urey, and Urey said, okay, send the paper again to Science. And Stanley had to write a telegram to Jack saying, please return the manuscript mailed March 13 by Harold Urey, entitled A Production of Amino Acid, etc. A mistake was made in sending it to you. Regret this inconvenience to you. Stanley was sort of embarrassed by the whole situation, but then the paper was sent back to him. He resubmitted it to Science, and finally the paper was published on March 5th, 1953. Rapidly. Stanley was absolutely delighted. But Stanley was a very timid person. He was very, very shy. And all of a sudden, you see a 23-year-old graduate student who is under the spotlight with an enormous attention, not only from scientific journals, but also from the general press. And it was very difficult for him to cope with the situation. So Stanley begins having doubts about how convenient it will be for him to develop a scientific career. And he decided to shy away from Science and to devote himself to become an artist. And he contacted his cousin, a wonderful woman that I had the chance to speak at length for several hours some time ago. She was a painter. And she instructed him to try to do some landscape painting. Stanley made this landscape, And believe me, the reproduction is much better than the original. He showed it to her, to his cousin, and she said, Stanley, go back to science. I think that you better go back to science. So he contacted again Harold Urey, and Harold Urey told him, it's very clear. You had an original mixture of methane, water, ammonia, hydrogen, and after the action of electric discharges, you have the synthesis of amino acids. What you have to do is to explain how do you go from methane, ammonia, hydrogen, water to amino acid, hydroxy acids, and urea. You have to find out the reaction mechanism. And Stanley then decided to repeat the experiment and follow very carefully the course of the reaction. And in doing so, he realized that, okay, here you have a diminishment of the concentration of ammonia, you have the increase of amino acids, but you also see hydrogen cyanide, which is a highly reactive compound, and aldehydes, which are also quite reactive, that were not present at the original mixture. How they come up, their concentration, and then they go down. And here Stanley realized very rapidly that he could explain the actual mechanism of reaction, which is the so-called Strecker synthesis, described in 1850 by Harold Strecker in Germany. So Stanley proposed then that you have aldehydes that result from the decomposition and reaction of water and methane, reacting with ammonia, reacting with hydrogen cyanide, the outcome of the reaction between methane and ammonia in a reversible reaction that leads you to the formation of amino nitriles, and this is reversible. And then you have the hydrolysis of the amino nitriles that will give you the amino acids without any problem. And he came out with a scheme to explain the presence of amino acids in the primitive Earth. The first part of the reaction will take place in the atmosphere during storms, and then when you had the sparks, electric discharges, and so on, lightning bolts, and so on, they will promote the reaction, and then the water falling down will actually hydrolyze the amino nitrile, and you will have the irreversible reaction, hydrolytic reaction, to give you the amino acids. And Stanley was perfectly happy with this. He published basically this as the outcome of his PhD thesis, and graduated without any problem. So the story goes. Then about 10 years ago, 12 years ago, I was doing a sabbatical period in Stanley Miller Lab. And once he came in, not so early, and he was showing me a letter that he had received from the Museum of Natural History in London, in South Kensington, offering to buy from him, to purchase from him, some samples of the original experiment, because I wanted to show them in a permanent exhibit. And when Stanley told me the amount of money they were offering and so on, I asked him, will you sell them the samples? You don't have any samples. And the answer was, of course I have samples. So we went into his office. He climbed over hills of PhD thesis, books, photocopies, and so on, produced a box that he kept in a bookcase. And in the bookcase, in that box, he had a sample of every single experiment he had done for the past 50 years. So here you have the description of the actual experiments, where it was published, Biochemical Biophysica Acta, the page in his laboratory notebooks, and the samples of every single experiment, which is absolutely amazing. And then he produced from one of these boxes the actual sample of the original experiment, which I found very moving. So I asked him, can I make a photograph of it? Of course. And then, do you mind if I show it to my students, to my colleagues? Of course, no problem. And then I told him, Stanley, what are you going to do with this material, which has a tremendous historical value? It has to go to the right hands. And then Stanley, we were quite close, in a very generous way, told me it will go into the right hands, and gave it to me, which was wonderful. So I was going back and forth between San Diego and Mexico all the time to check how my own lab was doing. So I took the sample, eventually just put it in my jacket, and went through customs without any problem. And a few years later, Jeff Beda, who was the second student of Stanley Miller's, second graduate student of Stanley Miller, and I were invited to give a lecture in Texas, in Austin. And the night before, we were comparing our slides, and Jeff Beda who has a very good geochemical, well, he now retired, but had a very good geochemical lab, asked me, what is this image? I told him the actual origin of the sample, and then he told me, why don't we analyze it by the contemporary analytical tools, which is a wonderful idea. Now, you can picture that going with a sample from the USA into Mexico is quite easy, but try to picture a Mexican going into the United States with a white powder of unknown composition going through customs. So after agonizing for a while, I decided to write a book, to buy a book on anthropological issues. Jeff Beda and a wife is an anthropologist. I cut a space in the inner pages of the book, put an aliquot of the sample, and send it by DHL into San Diego. And once the sample was there, I flew into San Diego. I sent a letter inside the package describing the material, just in case we were caught by the security officers, just to explain everything. So once the sample was in Jeff's lab, I flew to San Diego, and we decided to analyze it using the contemporary analytical tools, which are quite extraordinary. And in fact, we were delighted because we did non-acidic hydrolysis of the sample. We found the peaks of D-serine and the alanine, which are not found in contemporary biological system. And when we did an acidic hydrolysis, we found again the peaks without any problem, and beta-alanine, which was an indication that the sample was not that contaminated at all. And of course, the analytical tools available now for chemistry, organic chemistry, are quite extraordinary. So we decided to actually publish those results. But then we discovered something that is hardly known. Stanley Miller did not only the classical experiment, but he also did what we decided to call the volcanic apparatus. And Stanley Miller wanted to simulate the prebiotic synthesis of organic compounds in volcanoes in the primitive Earth. And the idea was to put here something equivalent to a pipette, a Pasteur pipette. So the water vapor will come out at high pressure, mixed here with methane, ammonia, and nitrogen, and with the electric discharges, and see what happened. He did not publish this because his fellow students told him that there are never lighting rods in a volcanic eruption, which is wrong. You may have seen The Last Day of Pompeii and those movies, and you always see electric discharges associated with volcanic eruptions. And there is a lot of geological records that this is the case. But Stanley paid attention to his colleagues and decided not to publish the results, which is a pity because the experiment is quite extraordinary. Here you see how Stanley Miller handwriting deteriorated as time went by. And here you see in his notebooks how he actually was experimenting with different diameters of the tube and so on. And when we did the analysis, we were absolutely surprised because, as you can see from the chromatogram of the results of the volcanic experiment, not only the diversity, but also the amount of organic compounds was much greater. So the lesson is, don't listen always to what your fellow students will tell you, because they may be completely wrong. So we decided to publish this. And another issue that we had to face is how realistic are our simulations when trying to understand the conditions of the primitive Earth. We don't have a sample of the primitive Earth. We don't. But we do have samples of meteorites, which are the building material of the planet. So we asked NASA for a piece of the Murchison meteorite that fell in Australia in 1969. We analyzed the sample of the meteorite looking for aliphatic amino acids. We then repeated the Miller experiment, adding CO2 and hydrogen sulfide. And you can see in blue the distribution of the aliphatic amino acids compared to the same amino acids that we found in a simulation experiment. And you can see that the correlation is on qualitative and quantitative terms quite significant. So we were quite happy, absolutely delighted, and say, okay, our experiment is a good model of what may have happened in the primitive Earth. It suggests, but doesn't prove, that there was indeed a primitive reducing atmosphere on the primitive Earth. And then we decided to publish the whole results in a science paper that actually, very much to my surprise, I have to say, caught the attention not only of our colleagues, but also of the newspapers. I myself, apart from what happened with Jeff Beta, I had to give some 15 interviews to newspapers and people from the news from all over the world, because they were absolutely enticed with the idea that you have sort of a chemical faucet of a famous experiment. And you can see here, compared with glycine, which is always the most abundant amino acid in a prebiotic experiment, you can see the diversity and the amount of the different compounds that we were able to analyze using contemporary techniques. I should add that not only did we analyze the original samples, but we repeated the experiments that Stanley had done, and they were absolutely consistent. But there's something there are many lessons to be derived from this analysis, but there is a couple of points that I would like to underline. There you are. Okay. Valine is an amino acid that we found in protein that has 5 carbons. When we do the experiments and we analyze the meteorites, we found not only valine, but also isovaline and norvaline, which also have 5 amino acids. But if you look at the abundances, both in meteorites and in simulation experiments, you see more isovaline than valine as an outcome. But we never see isovaline in proteins. So that is telling us that there was a selection due to reactivity, physical stability, structural restrictions, whatever, that did not allow isovaline or norvaline to be incorporated into proteins. And valine, yes. So clearly, one should picture that the prebiotic soup was extremely complicated, no doubts about it. Cells with RNA, proteins, DNA are extremely complicated, but the composition of cells, of extant living beings, does not reflect exactly what was taking place on the prebiotic soup. In other words, there must have been a tremendous amount of processes, as yet undescribed, that not only led to the first genetic material, to the selection of chirality, L amino acids, Z sugars over what was a racemic mixture of some amino acids after others, and so on. So clearly it was not a direct, life was not a direct outcome of the prebiotic soup. And we published this, we were quite happy and so on. And I remember perfectly well that once I was flying to Switzerland, and on the way, I received a message from a very dear friend, Dave Deemer, who also works on the origins of life. He works on membranes, and asking me, Antonio, you must read right away the latest book by Dan Brown. I have to say that Dan Brown is not my favorite author. I still prefer James Joyce or Balzac, but he strongly advised me to, as soon as I answered him in the plane, well, I'm about to land in Zurich. As soon as you land in Zurich, you must read the latest book by Dan Brown. So I did so. I was a little bit dizzy after a long flight, but I went to the bookstore in the airport, and there was a book called Origin, which was the one I was advised to read. So very rapidly, I didn't read the entire book. I'm not so enticed by this kind of literature, but I did found that Dan Brown, actually on page 91, actually on chapter 9, describes the following. It's experiment about, it's a novel about a character in Barcelona that actually tries to synthesize life. And he doesn't end well as he should. I mean, he ends up, I will not tell you how he dies, but in any case. So look what it says. Edmond Exal. However, as many of you may know, the Miller-Urey experiment failed. It produced a few amino acids, but nothing even resembling life. Miller and Urey eventually abandoned their experiments. The religious community breathed a sigh of relief, and the scientific community went back to the drawing board. He paused, and a amused glimmer in his eyes, that is, until 2007, when there was an unexpected development. And the novel goes on. Edmund now told the tale of how the forgotten Miller-Urey testing vials had been rediscovered in a closet at the University of California in San Diego after Miller's death. That's not true. He gave them to me. Miller, and it was not in a closet, they were in a bookcase in his office. Miller's students had reanalyzed the samples using far more sensitive contemporary techniques, that's true, including liquid chromatography and mass spectrometry, and the results have been startling. Apparently, the original Miller-Urey experiment produced many more amino acids, that's true, and complex compounds that Miller had been able to measure at the time. That's quite true. The new analysis of the vials even identified several important nucleobases, the building blocks of RNA and perhaps eventually DNA. And that's untrue. We never found purines, pyrimidines, not even sugars. That's absolutely untrue. It was made up by Dan Brown, which is fine. I mean, that's a privilege of a novelist to invent things. And actually, it's something, not a privilege, but a mistake that some scientists make. We looked for them, we never found any components of nucleic acids. The actual components of nucleic acids were synthesized brilliantly in 1960 by Joan Oró, who was able to demonstrate that hydrogen cyanide polymerizes and can give rise to adenine. That is true, but not what Dan Brown was saying. So, but all of a sudden, you can actually look at the book, and our papers are referred to as a novel and so on. My only hope is that if he eventually gets the Oscar award, that I will be invited to join Julia Roberts, of one of those characters in the red carpet. Okay, so let me now remind you of the actual scientific atmosphere in which Stanley Miller did the experiment. In 1953, he published the results of the outcome of the experiment, but it was published 3 weeks after Watson and Crick produced the double DNA helix model of DNA. It was quite an extraordinary year by all accounts for science. But clearly, at first, the two fields were absolutely separated. What joined them, in a way, was the discovery of the synthesis of adenine by Joan Oró. But at first, they were basically separated. And it can be said without any problem that during the first 20 years, during the 2 decades following the Miller experiment, our attempts to study the origins of life, that was when I entered the field in the 1970s, scientifically, people in the origins of life were working on the fact that evolutionary biology had become an established field of research. People doing evolutionary were no longer diminished as purely speculative thinkers. People were very much engaged in the unraveling of the details of DNA replication and how proteins biosynthesis proceeds, and the creation of NASA, which eventually led to this extraordinary level of sophistication with the many satellites that we have exploring the moon, Mars, and other bodies. But there was also the socio-political context that was basically the outcome of the Cold War tensions, which is exactly the years in which Stanley Miller published his experiments. Just to summarize this, one can say that in 1924, between 1924 and 1929, Oparin, Haldane, Lipp, and Harvey, but especially Oparin, published the idea of an heterotrophic processes of the origin of life. He was based on the extraordinary 19th century tradition of mostly German chemists that had synthesized urea, sugars, amino acids, a number of amino acids, but Oparin very brilliantly reinterpreted these results and uses as an example of what may have been the conditions of the primitive Earth. In 1953, Stanley Miller did the famous experiment. In 1961, Joan Oró published the outcome of the polymerization of hydrogen cyanide to give adenine, and this led eventually to the so-called RNA world model of life that we actually support today. So this is basically a scientific context. But what happened in sociological terms? The first thing that I had to say is that I missed completely the chance to ask Stanley Miller about the kind of pressures that he felt as a young student with no permanent position in an atmosphere that in the United States was extremely anti-communist, anti-socialist, and so on. It's very difficult for us to understand this properly, except with the fact that you might see in some places how you have this Islamophobia or you have this anti-Semitism in certain areas. When Stanley Miller did his experiments, the three main authors that he relied upon were, of course, Oparin, who was a Soviet scientist. He was never a member of the Soviet Communist Party, but he was quite close to the Soviet establishment. You can see in his papers how he relied on the ideas of John D. Bernal, an extraordinary character, one of the most significant crystallographers, physical and chemical scientists of the 19th century, the advisor of Rosalind Franklin, of Francis Crick, and so on, who was a very militant member of the British Communist Party, and Harold Urey, who was not a member of the American Communist Party, but who had refused to declare when he was called by the McCarthy Commission of Anti-American Activities. So you have a very shy student who was always very open-minded, but you have a very shy student who was actually publishing a paper in a society that's not very keen on evolutionary biology, on evolutionary thought, and basically under the intellectual influence of leftists, if you wish, scientists. In fact, Stanley eventually received the invitation to go to the Soviet Union, and you will see what came out of this. I never asked Stanley about what his feelings were, but I can tell you that in 1953, and I have seen the letters, Harold Urey was telling the American Army that they should not recruit Stanley to go to the Korean War, because even though he was single, he was nearsighted, and he wasn't doing beautiful work, and he should be able to remain in the lab. Those are conditions that are difficult for us to picture nowadays, but they were exactly what was going on. Then in 1956, when Stanley had already finished his PhD and he was doing a postdoc at Caltech, he got a letter from Oparin. The copy of the letter is in the archives in UCSD, inviting him to go and present a lecture at the first Moscow meeting on the origins of life. So Stanley got the letter, and he was very keen about going. Oparin insisted, but he wasn't sure if he should go. And he wrote a letter. I have never found a copy of the letter, but I did find a copy of the letter that Harold Urey sent him, advising him about the invitation. And let me just show you the last paragraph. To Stanley, from Harold Urey, I do not know how to advise you. I think each of us must make his own mind about this. The nuclear scientists, the American nuclear scientists, went some time ago, and if they let the nuclear scientists go in the United States without stigmatizing them, I should think that innocent people like us might also go, but one never knows what a McCarthy will do in the future. It is a very sad situation. And there was a McCarthy. Recall the politics of Mr. Trump, for instance. So Stanley was left without a clear piece of advice from Harold Urey, and he decided to apply for money from the American government to go to the Soviet Union. So he sent a letter in 1956 to the American International Science Foundation asking for money. He was advised by the American Science Foundation to ask the people from the Department of State for permission and money to go. And then he got a letter not from the State Department, but from the Atomic Energy Commission telling him, okay, you will receive the funds, you will be able to go, but upon your results, you have to report any significant changes in your itinerary. You cannot avoid discussing with people about restricted data. And as soon as you come, you must still ask, what did you see? to whom did you speak, what was your whole experience, which is somehow equivalent to the situation of your professors, my colleagues, under Franco regimes. It was basically it was really the McCarthy era. Now, Stanley finally received the permission and the money to go. And Stanley Miller, being a Stanley Miller, he decided that he should read as much of the Soviet Union as possible, or as Russian culture. So he went to the library, and asked for a novel. Let us say Anna Karenina. And the librarian lent him Anna Karenina. When he finished reading Anna Karenina, he went back and asked for another book, whatever you want. It was a novel. Pushkin, let us say, whatever, Eugene Onegin, if you wish. So he was lent the book. When he finished, he went back and asked for another book of a Russian author, Crime and Punishment, if you wish. And then the librarian told him, Stanley, this is the third book on Russia that you're taking out of the library. I'm afraid I will have to report you to the FBI. So this is telling you the kind of a scary atmosphere that was at the time. And keep in mind that Stanley was always very shy. So I can hardly picture, even though I knew him quite well, I can hardly picture the tensions, the pressure he must have felt. But eventually he went to the Soviet Union. And when he went back, he was visited by 2 government officers, and he was able to cope through the questioning without any problem. For instance, he told me that when they asked him, how was the atmosphere in Moscow? And the answer was very hot. The air conditioning was not working. So he was doing all these kind of tricks, but eventually I think that we will be able to access to the original reports from the FBI in the USA files. Now, having that said, the question that we can now legitimately ask nowadays is, are these kind of experiments valid models of prebiotic synthesis? The primitive atmosphere, this is a consensus now among my planetologist friends, is that the primitive atmosphere may have not been as reducing as assumed. It is that mostly CO2 and not methane may have been the dominant carbon species. Well, we decided to address this issue, and in fact, the original Miller experiment used methane, ammonia, hydrogen, and water. We repeated the experiment with CO2, nitrogen, and water, and we were able to synthesize amino acids without any problem. We found that it's an issue of keeping the proper pH. Once that is done, you can synthesize amino acids without any problem. We have not done the experiment looking for bases, lipids, sugars, or others. But that clearly is not an issue. And this kind of experiment in which you put all things together is what we call in prebiotic chemistry one-pot reactions, or in Spanish, reacciones de una sola ollita. Se mezcla todo, a ver qué pasa. But a very ingenious possibility has been developed by John Sutherland, namely, perhaps since different organic compounds are formed under separate experimental conditions, how compatible are these conditions with the so-called RNA world? Sugars are not easily polymerized, and then you remove them from the reaction schemes. So what John Sutherland and his colleagues came up with the idea of the so-called system chemistry, namely that you analyze all the different kind of prebiotic systems that we have and use any commonalities among them with the intermediates or the results to link the subsystems. For instance, you can synthesize a number of compounds. In a way, you can then use glyceraldehyde as precursors of lipids. You can actually use the processes that will lead you to amino acids to produce derivatives of hydrogen cyanide, or you can use them to produce other amino acids using metals and so on. So this has been done quite successfully. And for instance, this is a beautiful paper by Matthew Proner showing that since the traditional way of synthesizing cytosine doesn't work, well, perhaps we can use this number of different systems to make them interact and found different intermediates and then end up with activated cytidine. So I could say that right now, Prebiotic chemistry is divided into one-pot simulations or multi-pot prebiotic simulations, muchas ollitas. But the problem is that you produce one compound under given conditions, take the intermediates and put them under different conditions, take the other products and put them under different conditions, and that is slightly artificial or can be very, very artificial. So in a way, we have now two kinds of approaches to prebiotic chemistry. We have the Miller-Urey one-pot simulations that can synthesize sugars, amino acid, purines, etc. These are high energy, non-selective chemical processes. These are normally one-step productions of a single class of biomolecules. We have typically low yields of some additional compounds and the formation of a very complex polymer whose nature we have lots of problems describing. The so-called system chemistry approach developed by John Sutherland is that you have the synthesis of precursors of ribonucleotides, amino acids, lipids linked through what you can call cyanosulfidic chemistry. And these are low energy processes. They are led by the inherent reactive favor chemistry. You have multi-pot, diferentes ollitas, model of simultaneously interacting chemical subsystems. It requires the involvement of non-prebiotic reactions like the presence of copper, and this requires several metallic cations which were not clearly present in the primitive Earth. So there is a considerable debate in the community about which model we should favor, but regardless of these issues, regardless of this problem, what is perfectly clear is that this is all the intellectual, scientific inheritance of the original Miller experiment. And I could end the seminar here, but a few years ago, by pure chance and intuition, I was in the FENAC in Madrid and saw a book that attracted my attention by Harry Mulisch, a Dutch author that has since died. The book is called The Procedure. It's a book that every single young scientist and every graduate student in science should read, because it describes the kinds of sacrifice and the inner tensions that you suffer when you want to make a scientific career. Should you follow your heart and keep with your companion, or should you leave your girlfriend or boyfriend and devote yourself to go to another place? Are you willing to extend the hard times working for a PhD thesis, the hierarchical system, the lack of funds, the having to move from a different place? And it's an extraordinary book that I very strongly recommend to you. And when I saw the book, really I was just attracted by the cover. I found that the book actually has as a character Stanley Miller. And when I showed it to him, he was absolutely surprised. So the character says, I was born in 1952, but my philosophical birth took place in 1953, the crucial year in microbiology. It was then that Watson and Crick constructed their model of DNA molecule, the famous double helix. In my philosophical year of birth, another remarkable experiment was conducted, in which I'm also closely connected. In the 1940s, provoked by a Soviet scientist, Oparin, there was a debate about the origin of life on Earth. His motive, of course, was, after Darwin, to eliminate God even more definitively, which is not really the case, but still. And he goes on. Everybody agreed that a major role in the origin of life was played by 20 amino acids, the building blocks of proteins, but how had they originated? An impudent student, a wonderful phrase, an impudent student, Stanley Miller, had the kind of brilliant thought that doesn't occur to a sensible person. Let's try it, he said. With the kind of chemical equipment that every inquisitive adolescent has in their attic, he simulated the presumed condition of the Earth atmosphere at the time with hydrogen, methane, ammonia, lighting, etc. And he made the sparks fly through his mixture. And what do you think? After just a week, he was able to demonstrate the formation of all kinds of essential amino acids. I myself cannot complain with my LBO. After all, even the Pope has warned mankind against me. But that pure, elegant simplicity of Miller experiments awakes in me a respectful admiration. And I think that you will agree with me that for a young scientist, a student with no permanent position, the pure elegant simplicity of the Miller experiment does awake in many of us a respectful admiration. Thank you very much.
Questions, so please. Okay, Domenic.
Thanks for the nice talk and also entertaining. You mentioned briefly chirality, and many of the amino acids have chirality.
Yes.
So when you produce in those experiments, I would think you produce in both chiralities.
Yes. All the prebiotic simulations done so
but the
far.
reactivity is the same. So
Yes,
then,
exactly.
so then how?
There were claims by Sandra Pizzarello that you could find in some meteorites actually a non-racemic mixture. And but so far we don't have a simple explanation. Some of my theoretically oriented colleagues, mostly theoretical physicists, actually say, well, perhaps it was a chance event in a mixture that by chance had accumulated mostly L amino acids over the amino acids. Other people say, well, perhaps we had very strong electromagnetic irradiation of some samples that favor the L amino acids over D. But the fact is that we also need to explain the accumulation of D sugars over L sugars. And so far, the best experiment I can think of was reported by Sandra Pizzarello and her colleague, Art Weber, that they did a bootleg of synthesis of sugars, added proline. If they added L-proline, they synthesized the sugars mostly, or if they added D-proline, they synthesized mostly L sugars. But that's about the state of the art. It's an open question.
Okay. Jaume, you have
It's a troubling open question, I should say.
Thank you very much for this very nice lecture. I was curious about this variation of the experiment where we introduce a volcano. So I have no background in chemistry. So I'm curious, though, to see how this favors the synthesis of certain compounds.
The only explanation that we can provide, we being all the co-authors, is that if you have water vapor, the reactivity of water vapor is much greater with the intermediates of the amino acids that are being formed, that if you put those intermediates, which are in themselves highly reactive, in liquid water. We did see a number of amino acids that had the hydroxyl group, the OH group attached to them. So that is consistent with the possibility also that perhaps the electric discharge was decomposing water and producing lots of hydrogen, hydroxyls that were reacting with the precursors of amino acids.
Okay, another question, Javier.
Yes, so you mentioned this meteorite, Murchison
Yes.
meteorite, yes. And if I understood correctly, you use this because this is kind of a source of the original composition of the solar system. But I thought that the interesting thing about this meteorite is that you can actually find amino acids there. You don't have to synthesize them, not
Right.
So they are there already in the meteorite.
Yes. That is conducive to the conclusion that the meteorite may be a piece of a planetesimal, a larger body that broke down, and that planetesimal actually collided with other ones and eventually formed, let us say, the Earth. Now, how do you form the amino acids in the parental body? And so far, the only or the best processes that I know that has been proposed assumes that you have in the planetesimal, you have a heat source of decaying organic compounds, of the decaying radioactive material, that it heats up the water that may have condensed over the surface of the planetesimal, reacts with ammonia, hydrogen cyanide, and so on, and you have a typical Strecker synthesis. This was published by Jeff Beda and Stanley Miller about the late 80s or the early 90s in a paper that, no, it's so far the best model. The other possibility that we think of is feasible is that you might have in the primitive solar nebula, you might have Fischer-Tropsch-like reactions that could synthesize the compounds under high pressures, quenching a lot of activated compounds, a lot of activated intermediates, and that could give rise to the compounds. And a third possibility suggested by Akiro Fukara from the Japanese group, she proposed it 4 months ago, is that one may have on the surface of the planetesimals, the parent bodies of the meteorite, we might have some interstellar organic molecules that become attached and that were present in the solar nebula as it was condensed. But clearly the compounds are of extraterrestrial origin, and that is strongly supported by the analysis of the Ryuyu asteroid reported so far by the Japanese colleagues, where you find a number of amino acids, proteinic and non-proteinic amino acids, racemic mixtures, and you also find uracil and vitamin B, which is a derivative of uracil. We are all expecting the results of the Bennu asteroid by the American group.
Okay, yes, Mike.
Thanks, it's a very pleasant talk. My question, I'm not sure if my question is relevant or not due to my ignorance, but they play any role, the galactic cosmic rays, on all these processes, or are there any kind of study that has evaluated the influence of the galactic cosmic rays?
Yes, some people have argued that the asymmetry that we see in the chemical mixtures from meteorites and asteroids could actually be the outcome of the asymmetric irradiation of cosmic rays. But it's an idea. There are a few experiments, but it's not a subject I will suggest to any graduate students at the time being.
Any other questions? So I also have a question I wanted to ask you. That we see, we learned how these basic molecules can be synthesized in the primitive Earth, also in some parent bodies of these meteorites. But how do you envisage continuing on this route to, I guess, the missing thing is how do you go from these basic molecules to form the first organism, right? Or was the first cell something similar to the archaea or prokaryotes, or what is it? So how do you see the progress in this?
That's a different lecture. And I will actually speak about that very briefly tomorrow. But the idea is that once you have monomeric material in the primitive Earth, you have a number of microenvironments, for instance, small ponds that can evaporate, or you have the presence of a number of very active organic compounds like cyanamide that can actually promote condensation reactions, and you can have the coming together of amino acids or their precursors to give you a small peptides and a small genetic polymers. Now, the actual composition of those compounds is, of course, open to debate. But I will say that in a more general way, I will say that we approach the origin of life by two means. On the one hand, we have extant living beings, and we try to go back far back in time as possible, picturing a very simple biological system. And we do have some success in that. For instance, my friends working in ribosomes cut off a number of proteins. Ada Jonat, she has shown that in the inner, in the sanctus sanctorum, if you wish, of the ribosome, there are no amino acids, there are no proteins. So you can picture a purely RNA-made ribosome. One can picture simpler amino acids or simpler peptides. But that's what we call the top-down approach. The down-upwards approach will involve in general was very keen on that, trying to simulate liposomes, putting together different components, see how you can come out with selective process that favor one type of liposomes over the other. The work of Irene Shen, she's in UCLA, is beautiful in this regard. So what we try to work, this is actually an analogy that was used by Oparin, remember, who was writing at the time of the Soviet Union. He was describing two sets of workers going through a mountain until they found together and they joined the tunnel. So that's basically what we try to do.
Okay, thank you very much. Okay, so if I don't see more questions, one thing I'd like to mention to finish is that this great colloquium by Professor Lazcano was possible thanks to the collaboration of our Institute of Cosmos Sciences and the Catalan Society of Physics. And well, I just would like to encourage more of you to become members of the Catalan Society of Physics. And if you would like to ask anything, just let me know. You can find the website and so on. So just to finish, let's thank again Antonio Lazcano for this excellent colloquium.
¿Estuvo bien? Qué bueno. Se quedan todas las diapositivas, ¿eh? Aquí